Welcome to Under the Microscope Biology Magnified. Join us in our cosy biology corner and get ready to smile, laugh and learn about the amazing scientists doing good and changing the world through their research. Whether you're looking for ideas for a new project, a career or just for fun, welcome. It is lovely to have you here. We'll have some long-form interviews of researchers, scientists and professors about how neurons develop, to pandemic prevention, to cancer cures and all the way to genetics. Or you can join us for a short snippet of a bio bite. Be kind, lovely and stay curious. Hello curious people and welcome to Under the Microscope, where we are exploring the biology research making the world a better place. Join us as we discuss articles, papers and interview experts to explore the cutting edge of biological research, or take a quick bio bite to go about new scientific developments. Under the Microscope, Bio Bite 1. HIV, uninvited intruder or mystery medicine? The story of HIV has been one that shaped human history, from our understanding of medicine to how our health system functions to social perceptions of disease. It has been intertwined with the deadliest diseases known to man, such as smallpox, malaria and tuberculosis, and has also now been associated with technologies that are injected by 200 million people worldwide every day, and even what nearly every single person alive used extensively during COVID testing. Today, we are looking at the mystery biological features that make HIV such a perfectly engineered threat, some potential cures, and perhaps how HIV has been used to our advantage to enhance the future of biology. HIV, or human immunodeficiency virus, is a microorganism that is associated with an immune system disease, and today we're going to look at 4 cunning ways it causes this damage, from silent symptoms to genomic hijinks to commandeering our cells and evading our immune systems. But first, the story of HIV in the eye of humans tends to be told starting as a surge of symptoms that were short-lived and varied, that then ended in severe infection and death. Our story, as most historical stories, start with discrimination and an anti-LGBTQ rhetoric. Communities would be devastated by these endemics, which at the time were noted as falling in certain minorities, and so the stories of what the disease was and what caused it mirrored this great unknown. Rather than a complex understanding of the mechanism of infection, or that it was actually a hidden and long-lived disease, HIV was commonly seen only in the later stages and was nicknamed the gay disease or the devil's incarnation, and believed to be caused by immoral behaviour or straying from religion, particularly related to sexuality. Although our current understanding of this pathogen does corroborate that some groups may be at higher risks, HIV itself doesn't discriminate, and anyone, anywhere, at any time can be exposed, from babies to the elderly, and it is up to us to ensure that we can protect them. Judgment and social taboo is one of the reasons that despite affordable, effective and available treatment, most parts of the world still have active HIV infections, or some communities may still refuse treatment or diagnostics. HIV is no longer a death sentence, but if we don't discuss it and seek to understand it, then each silenced patient, each alienated victim, each scientist who fought against the discrimination, each person who hugged and held those who were ostracized, and each individual who had tragically lost their lives will have done so for nothing. Today we are exploring the reality of HIV, how it happens, how it develops, and how we can change the perception of a virus that is no more immoral than the common cold. HIV is now known to come in 4 stages, and this is the first of our biological features, silent symptoms. When you initially get exposed, commonly by exchange of bodily fluids such as blood, reused needles, sexual relations, or in utero to a developing fetus, the symptoms mirror a minor cold or flu, maybe some mild fevers, some lethargy, but nothing out of the ordinary. It's unlikely anyone would question if they were harbouring a potentially fatal virus after a few days or a week of normal symptoms that then went away on their own, and you wouldn't follow up for testing or treatment in the early stages. Now, I don't want to scare you. HIV is so much less common and dangerous now, even if it is caught in later stages. But if you do have some queries of exposure, such as doctors, frontline healthcare professionals, or those at high-risk groups, a simple and cheap test can detect presence, and post-exposure prophylactic therapy can be taken to stop the infection from taking root. However, once you do enter the seroconversion stage, the HIV has started taking hold of your body cells, and this is where the complications start to arise. After this minor cold or flu episode, for a few weeks, months, or even years, you won't notice anything is awry. Your body is producing HIV virus and is being taken over slowly, but it's gradual and silent, waiting to ambush you, and you're likely to continue your daily activities, potentially spreading the virus or impairing your health. Barrier contraception, PrEP for partners, and other protective antiviral therapies could be taken, and at this point it means you would have no significant immunosuppression and wouldn't be passing on the virus, even to a developing fetus. However, by the time that genetic changes occur, it may be harder, but not impossible, to treat and manage. Plus, by this point, the symptoms will become evident, and it's only then where typically diagnosis occurs. The HIV virus works by binding to the CD4 receptor on your T cells. These are a branch of your immune system. These specialist white blood cells are your defense mechanism. They're meant to identify if your body cells are different in some way and tell them to self-destruct. All of your cells have an antigen, or a protein coat, on your body cell. This is how your body can recognize, this is me, this is me, this is different, so it's dust or pollen or a bacterium, or maybe it's my own cells that have been hijacked by a virus, have mutated into a dangerous state, or might be precancerous, so the T cell will just bind to that slightly changed protein coat and tell that cell, shut it down. This means that it only leaves healthy working cells behind. However, when HIV targets these T cells, it is no longer possible for the T cell to then bind to your own atypical cells. So HIV viruses can be continuing to be taken in by your body cells, hijack them, change them, whilst the T cells just have to sit and watch, unable to intervene. Of course, the first biological feature, a silent threat, is only then magnified by the second and potentially most impressive feature of all. The central dogma of biology basically states, and I am massively oversimplifying here, that genetic information is passed down to code for all the functional proteins and polypeptide sequences needed for life, so information can be inherited. In your body, complex DNA codes for proteins, but the DNA doesn't directly do this. Instead, it is transcribed and translated, which means it's almost changed into another language. Then, ribosomes, which are like little assembly units in factories, can build these little words of the new language into the proteins needed. The DNA is interpreted into the new language, RNA, which then can code for the chain of amino acids that make your proteins. This DNA code is just a sequence of bases, AT and CG. It's almost like DNA is this massive textbook, and when we make RNA, then we're just taking a few pages that we actually need to cram the night before a test, and these few pages is all that you need to code for the protein. Viruses and other organisms also have DNA or RNA. This can be double or single-stranded, but either way, when injected into a host cell, they use this instruction to manufacture the proteins they need. Usually, they can look like the RNA language that our ribosome factories are used to reading, so they just produce these proteins as if it's your own body coding for them. However, most viruses just have this simple code read and produced, but it's not inserted into your own DNA, it's not treated as if it's your own information, and then the virus will either leave or kill the cell, so your own genetic information of the host doesn't change. That is apart from HIV. As if waiting and biding its time for years wasn't enough, it also literally changes who you are. Or at the very least, it rewrites part of the DNA in the infected cell. Using two enzymes which are deadly, tragic, and also so beautifully evolved, called integrase and reverse transcriptase, it basically changes its genetic code into one that looks like DNA, so the host cell can pick it up and put it inside your own. This means with each cell cycle, each mitotic division, every second of every day, your DNA will grow and copy this code and put it into all your daughter cells. Slowly, all of your cells will be programmed to manufacture this HIV virus protein, and not just making it in the little factories, but it is now part of the giant textbook that codes for you. This makes HIV a lot harder to treat than just a single infective agent, and it's why antiviral therapies are lifelong and daily, since each cell division accumulates more cells that are making the HIV virus. Your somatic or body cells in you just keep making copies, and unlike editing an embryo that is developing or changing just a few genetic bases in just a few of your cells, the ability to either remove or neutralize the code for HIV in every cell of your body is a task that takes time and money and comes with way too many risks and side effects to be viable right now. Then come in sneaky features 3 and 4. This is how HIV commandeers our cells and evades sending out any red flags until it's too late. You see, most viruses will inject their information coding for proteins, have the host cell manufacture the protein, and then burst out to spread the particles. But usually your body starts to realize that something is wrong when there is just a scattering of dead cells. Plus, your normal body functions aren't working either because you don't have cells to produce your own body proteins. However, with HIV, instead of lysis or bursting, it does a process called blebbing. It also makes your own host cells manufacture the needed proteins and creates a clump near the edge of your cells, but rather than bursting forth into your body, it nips off a piece of your cell membrane and leaves as a mini package, whilst your cell still remains intact. This means it can continue making the viral proteins, it can continue passing on the changed DNA, and it doesn't immediately alert your body, since it's still manufacturing your own proteins and molecules, even if at a reduced load. Your body may be a bit more tired, but it's not acutely in distress, so you may not notice the deterioration. Then, sneaky biological feature 4 is linked to this blebbing, as the piece of your cell membrane that it encapsulated itself with allows it to mask that it is actually a foreign virus. All of your cells have an antigen, a protein coat, that has things like cell-cell signaling proteins. They basically tell your immune system, don't attack me, I'm your own tissue. Or at the very least, until you get an autoimmune disease. It is not fun. Do not recommend. Whilst foreign antigens such as dust, pollen, bacteria, or in my case, normal human foods that are definitely not trying to kill me, but my immune system clearly didn't get the message, do not get this defense and will be destroyed by your immune system. But since the HIV virus is encased in your own membrane with your own code of proteins, the immune cells just recognize it as itself and don't attack, allowing the virus to further run free. It's definitely looking like HIV-4 human 0, for now, but perhaps we are starting to level the playing field. For example, if HIV is so great at getting into cells and stopping T cells from attacking and destroying cells that aren't your own, what if we made HIV get into a cell and inject a payload of our choosing? For example, for those with insulin-dependent conditions, you may have noticed that your insulin probably no longer says of porcine origin, and that is because of HIV. Or at least it is because of the human ingenuity of scientists trying to turn this deadly weapon to a useful technology, where HIV genetic information can be exerted and integrated into a small yeast organism to instruct it to make protein, but instead of a deadly virus protein, to to make insulin. This means less risk of rejection and immune system issues, plus it avoids the cost, both ethical and monetary, of porcine insulin production. Perhaps a more cutting-edge application of inserting HIV with useful payloads is in animal or human trials, such as into the pancreas of a type 1 diabetic to make it produce its own insulin, cutting out daily external treatments and injections. Or maybe in the future deactivating your immune response, preventing development of disorders such as type 1 diabetes in the first place, or against immune attacks of transplanted organs, or even injecting these very organs with HIV-mandated technology to add hybrid genomes to reduce rejection post-transplant. The future sure does show that even the nastiest pathogens may hold the key to saving lives one day. Another amazing thing that HIV enzymes can do is create more of genetic information. Whether you're tracking genomes for conservation and breeding programs, you're doing ancestry testing, forensic analysis, identifying a disease, or if, like me, you remember sticking cotton swabs up your nose through the pandemic, it is likely that the tiny sample of DNA that you would have collected would have been replicated, usually using one of the HIV's enzymes, in a process such as PCR to be able to quantify and analyse what virus, bacteria or genome was present. I certainly am grateful that this invention means that we can do a quick and mostly painless swab, rather than a very long and intrusive blood test or filling up entire containers of much less pleasant fluids. So perhaps we can now begin saying HIV for humans 1? It may be optimistic, but many scientists have developed even better screening tests for asymptomatic stages and potential gene therapy trials. So maybe in the future, HIV wouldn't be a lifelong treatment that you always have to battle, but a 1 or 2 times pill that you can take to cure this deadly infection. Unfortunately, this isn't the reality as of yet, and with that we come onto the later stages of a HIV infection. Stage 1, or the flu-like seroconversion. Stage 2, asymptomatic and silent. Stage 3, short-lived progression of symptoms that may look like cancer or TB, such as heavy night sweats, fevers, unintended weight loss, severe fatigue. All of this then culminates in Stage 4, AIDS, acquired immunodeficiency syndrome. Here you are incredibly susceptible to any infection and other complications such as cancers. Even though it is now harder to treat than an earlier catch, it can still be reduced with modern technologies in the symptoms and spread by lowering the active viral load in your blood through daily tablets. And although it can take a toll for a lifelong commitment, it is quite an easy way to avoid a much more tragic stage that now means those with HIV should not, with advice of medical professionals, have to limit or change their daily life as much as previously thought. Before these modern technologies, any opportunistic infections such as a simple cold, a flu, chickenpox could mean a very tragic end, especially for someone with AIDS. But now, thanks to modern medicine, we can keep the viral load under a safe amount. Someone with an active but managed HIV infection can operate, do science, prepare food, work in sterile areas, and so much more. I really cannot stress enough that the way HIV has been painted is no longer the case, and this is due to the amazing scientists, researchers and dreamers who didn't listen to the very strong opposition, doubt and discrimination. I am so thankful to live in a world that continues to try to help others, and grateful to work off of the amazing foundations that scientists before me had made, such as identifying the viral cause of HIV/AIDS against the previous religious explanation. Public figures such as Princess Diana, who despite not being of a scientific background, shined much needed media attention and sympathy, and activists, educators, engineers, researchers, lab teams, clinicians, healthcare professionals, doctors, and so many more who changed the medical, scientific, and social narrative of HIV. Now, the 4 sneaky advantages that we have looked at are definitely responsible for a lot of the tragic deaths, illness and pain for victims of HIV in their community, and the silence, length, invisibility and then sudden nature of the infection means most treatment has been too little too late. And even now, with access to treatment, it may be expensive, or just not available, or difficult to keep up with the daily regimen, or worst of all, it could still be taboo. Most comorbid infections with HIV are opportunistic. They wouldn't pose such a big threat to a healthy individual, but with stage 4 AIDS, then the immune system is so overrun that it can't fight a simple cold. Vaccinations with live or weakened pathogens can be too dangerous, and immunosuppressants can then cause HIV numbers to increase. So individuals with advanced stage HIV may be in strict quarantine, isolated from loved ones, and rely on the health and safety measures taken of every member of the public they come into contact with. It can come with many secondary issues such as skin fissures, neuropathy, shingles, pneumonia, and also provides a breeding ground for hybrid transmission. For example, some tuberculosis strains mixed with HIV may mutate to one that is even deadlier and evade our medication, such as extreme drug-resistant TB, which is resistant to all 4 families of our frontline drugs and can only be wiped out with very heavy and very, very painful chemotherapy, which just kills all the dividing cells indiscriminately. This is why the disease has such a disparity in treatment and quality of life. HIV in a high-income country without social taboo and with access to healthcare means you can continue living your life as normal. Elsewhere, even in 2024, HIV can still be seen as a death sentence. However, we can also look to the future. If patients had a shorter course of medication such as gene therapy, if they could get regularly screened, if taboo was reduced and stigma and shame by communities were no longer enforced, and customs and cultures were respected in how information and policy around diseases was adapted, and if a doctor asking you if you'd like to be tested for HIV was as normal as being tested for strep, a cold, or COVID, then we may one day be at a place where HIV is thought of as a biotechnology to make medicine, a way to make transplants safer, a way to test for diseases rapidly, and just a story one day lost in the history textbooks about the brave men and women over the decades who had managed to overcome our own uncertainty, fear, and tragedy to once again save lives with science. And who knows, maybe one day you will be stood accepting your Nobel Prize as the individual who cured HIV once and for all, and rewrote a story of suffering and pain into one of medical miracles, happiness, and the future of biology. And that is a DNA wrap on this episode. As always, please do let me know if I muddled anything up. Science is constantly changing and I'd love to learn more. I hope you enjoyed hearing about the amazing biology that is changing the world for the better. Have a wonderful day and be kind, lovely, and stay curious.